Okay, so what we'll do is start with a little bit about our organizations. So Mahdi, can I hand over to you so that you can tell us a bit about UBC?
For sure, for sure. I'm just going to share my slide, then share screen. So Lesley, I think you need to stop yours. Yeah.
I'm gonna refill mine since you said it.
I'd like maybe to say to everybody to mute. So Lorenzo, or can you mute them directly? Ah, right.
Muted, yeah.
Thanks, Leslie.
Okay. So I'm just going to share. Okay, so my slides are coming. Okay, just going to switch this. Okay, great. So I assume everyone can see my slides now. Perfect. So yeah. Hi everyone. Good morning to the people in here in BC, and good evening to the people in Europe. Thanks very much for joining. Thanks a lot, Lesley, for taking the lead on organizing this. I really appreciate that. And it's great to see that researchers, faculty members, students, postdocs from the two groups are joining. And I hope that I'm sure that will be a very fruitful discussion. We'll have good presentation. Also, the discussion around an important topic related to microplastic research. So my name is Mahdi. I'm joining you from Vancouver in the province of British Columbia in Canada. And the people who are joining from our side are the members of this research cluster at the University of British Columbia called Cluster for Microplastics, Health, and the Environment. Should silence my phone. Okay. And this is our website, mp.ubc.ca. I encourage you to visit to get more information about our cluster. So UBC, University of British Columbia, is, as I mentioned, is in the province of British Columbia, and it has two campuses. The main one, the Vancouver campus, and just right at the Pacific Ocean, and the other one is in a city called Kelowna, about 5-hour drive, and it's called the Okanagan campus. So that's the name of the region, Okanagan. So the researchers in our cluster and are from both campuses of UBC. So this cluster, microplastic cluster, is a multidisciplinary, multi-campus research cluster. It initiated through an internal funding support from UBC, which is supporting collaboration among researchers at UBC on topics that are important, that have impact. And of course, microplastics has been identified as one of them. And that's why this research cluster has been formed. And the researchers, it's currently we have over 20 faculty researchers across from different departments at UBC that are part of this program. Some of them are currently heavily engaged in microplastics research. Some are they have related expertise that they can contribute to the research, and as the cluster is trying to expand and initiate new projects, of course their expertise will be leveraged. And of course the cluster is addressing the grand challenge that a lot of course, both the CUSP group and also us are working on, and it's in the news like that microplastics are detected everywhere, and really what are the impact, how we can understand it, how we can characterize it. And the overarching goal of this cluster is offering scientific insight and evidence to support corrective actions and policy development to reduce their impact and for building, of course, a more sustainable, cleaner future. And that's why we are, our clusters, or we are, our researchers and our cluster is trying to really work closely with different government departments at different levels, federal, provincial, and municipal, to really contribute to that aspect of policy development and facilitate that, support that knowledge translation, as well as, of course, communities who are and other some industries that are some kind of research organizations, nonprofit research organizations who are working in this field of microplastic research. There are three themes, human, that are currently, it was built around these three themes, and the researchers are working in one of these, or their expertise can contribute to different, and of course some of them are really, it's bringing the expertise of all of them together, let's say the policy. But human and ecological health, source type and pathway characterization, and policy management strategies and knowledge mobilization. And the research of the cluster each is contributing to one or several themes, research projects that are currently happening in the cluster. Examples of these ongoing projects are two of them are the one, and they're both funded through government programs. Different programs. One of them is focused on understanding the seasonal impacts of microplastic inhalation on health. The lead PI on this project, which I'm not sure if she's here, but Dr. Sepi Depakpur. And the goal of this project is focusing, of course, in the microplastics that could be found in air, and they're primarily focusing on the adverse, the health impacts of these microplastics. The other one, which the lead PI, Professor Maite Maldonado, which I saw her, she's in the workshop, is focusing on sources, sinks, and fate of microplastics in the Strait of Georgia, which is close by actually the region that Vancouver is located. And that one is really focusing, bringing together researchers from different departments, from ocean studies, chemistry, medicine, microbiology, and civil engineering. They're all working towards studying the source, transport, and fate of microplastic in the state of Georgia and its urbanized watershed. And they're also focusing, of course, on both experimental works, understanding the microbial degradation of microplastics, as well as they're developing predictive models to evaluate the transport of microplastics. And I think that's it. I want to keep it short. So this is the website of our cluster, and also this is my email. My role is as the coordinator of this cluster. So I would be happy to, if there's anything, please reach out to me. I would be happy to, for any collaborative idea, if there's any funding program that you think the two groups can apply together, please reach out to me. Feel free to reach out to me, and I can connect you to of course, the researchers who can offer complementary expertise or other activities, joint activities that we can do together. Thank you, Leslie. I'm passing this to you. Appreciate it.
Okay, thank you, Mahdi. Right, so for the benefit of University of British Columbia people who aren't familiar with us, can you see my screen? Yeah.
Yes.
Okay, so just a quick tour of the CUSP cluster, because all the information is available on our website, but just the important things. There we are. Okay, so who are we and what are we doing? Okay, so we comprise 75 organizations over 21 countries. There are 5 large-scale projects funded by the European Commission. 4 of them are 4 years, and then Aurora will be for 5 years. And you can see the names of the projects. There's Aurora, Imptox, Plastic Heal, PlasticsFatE, and Polyrisk. If you go to the website, you'll see all the links and the connections and the names. And also you'll see the individual focus of each project. So together we're going to contribute to the health relevant aims of the European strategy for plastics in a circular economy and the bioeconomy strategy. We'll also be contributing towards REACH restrictions on intentionally added MMPs to products, and we'll be providing new evidence for better preventative policies. So that's one of the joint aims as well, as to inform policy. Okay, so as mentioned, each research project is investigating a different aspect related to MMPs and health. And these include possible harmful impacts of MMPs on pregnancy and early life. That's the Aurora project. The relationship between MMPs, allergic diseases, and asthma. The impact of MMPs on the human intestinal tract and the immune system, and the development of a comprehensive measurement and testing program. Okay, the other thing is that we have a rotating coordinator role. So over a 1-year period, a different project is responsible for coordinating CUSP, apart from the final year where it will be 2 projects together. But throughout this time, the working group leaders and the members of each working group will remain the same. So the working groups are really kind of the linchpins, the cornerstones of our collaboration. We have 6 working groups. And so this workshop is, I suppose, predominantly aimed at 1, 3, 4 to some extent, organized by 6, but it'll be relevant to everybody in different ways. And maybe not necessarily your work within this specific project, but other work that you're involved in. So each working group has a representative or 2 from each of the 5 projects. We have regular meetings and we plan our strategies. Working group 6 is very much focused on collaborating, stakeholder engagement, and we try to collaborate and share knowledge and experiences with other organizations, including UBC, networks, clusters. This includes Momentum in the Netherlands, the Austrian MMP network, Nanosafety Cluster, Plastics Europe, Papillons, and it's ongoing. I've put some relevant links here so that you can follow up. The first one, that's to the page with our working groups. Second one is to sign up to our newsletter and any other updates. And then to follow us on Twitter and join our LinkedIn group. And that way you can keep up to speed with everything. And we'll try and do the same. Okay, so that's it, short and sweet. Okay, what I'd like to do now is just move on to our presentations. Our first presenter is, okay, I'm just going to stop sharing. There we are. Okay, so our first presenter, Florian Meirer, I hope I've pronounced your name correctly.
Yeah, that's okay.
Okay, so Florian has to scoot off for another conference. So we're actually really appreciating his time in delivering this. So Florian, I'll hand over to you and you can share your screen.
Thank you very much, Leslie. I will try to keep it nice and short, as you already said. And let me see, you should see something now, I hope. Yeah? You see the slides?
Yeah.
Yes.
Perfect. Thank you. Yeah, so thanks again for asking me to show something today. It's well, honor and a pleasure to introduce a bit what we are doing. I called it an update because why we just started with all these projects, right? So we don't have polished results yet, but focusing on the challenges and the things we do at the moment. And I chose to focus on force microscopy and tell you a little bit about what we do here with this method in Utrecht in the Inorganic Chemistry and Catalysis group. Now, before I start with that, you might wonder what is Chemistry and Catalysis group doing here. We are used to look at small particles in catalysis, specifically when we look at metallic nanoparticles in catalysis. But we have actually started to look much more into research into plastic. And our ICC group, Inorganic Chemistry and Catalysis at Utrecht, we would like to, very much in the spirit of what Lesley already mentioned of the circularity and circularity in chemistry, like to close this loop between making plastics. As you can see, we have several projects here on the making of plastics, working also with industry and the Dutch Polymer Institute on polymerization catalysis, so making the plastics. But we also care about the end of life of these products. I don't need to tell in this community that this is, of course, a problem. So we have a team, the Nanoplastics team, I like to call it here, working on the impact of the plastic in the environment. And if it's not properly recycled, and this is the third pillar in the plastic research in our group, where we focus on recycling methods, chemical recycling, but also pyrolysis, using catalysts to improve the recycling options and close, if possible, the loop, right? And then avoid the negative impact on the environment in the end of life. So today I will focus on the nanoplastics group. These are the guys who actually do the work. I'm just allowed to present a bit what they are doing. So we have we were lucky enough to be partner in 2 of the 5 projects of the CUSP cluster. So we have Adrian Duindam working on the Polyrisk project and Laura Sautendijk working on the Aurora project together with Laurens Mandemarker, our postdoc. Of course, we have a brilliant group of very talented and enthusiastic PhD candidates and master students here, as you can see. I also listed the other members of the nanoplastics team. We have 3 very enthusiastic and involved master students at the moment, led by our PhD candidates. And one of the projects we also look at related to the impact of nanoplastics, micronanoplastics, is looking at samples from the ocean and looking at how micronanoplastics, for instance, also distribute in the ocean. And that is why I also listed Claudio Pirar here. He's working at Utrecht, but not in our group, but in the Physical Oceanography group from the Marine and Atmospheric Research Institute. And he's going to simulate, or he's simulating flows or mass transport of micronanoplastics in the oceans. And we work together with him because, as I said, we are chemists, we are spectroscopists. We like to understand what happens to the plastic and use our methods to provide, for instance, additional information to Claudio to improve his simulation models. Well, let's jump in. What do we do and what are we trying to do with force microscopy? Well, I grouped the analytical efforts at the moment we have in our team and also in the Aurora and Polyrisk consortia into the guys who look at mass, right? So detect mass, and the particle counters. And we are obviously particle counters. We are looking at, we are trying to count particles. So hopefully not like Lucy here without keeping track of them. I hope it's not me ringing here. Sorry. No, it's not me. Someone, I hear a call. Okay, anyways. Okay, obviously it was me, sorry. So we are trying to keep track, not only count particles like Lucy here, but really trying to keep track of the particles. And we using force microscopy, we really focus on the nanoplastic side. There's already a lot of work done on imaging microscopy, counting micron-sized particles and larger. We want to focus on those that are really in the smallest size regime, where we are limited with visible light due to, of course, the diffraction limit. And we want to characterize these individual nanoparticles. So very obvious to everyone, but I just want to take a few seconds to remind everyone, nano is not micro. So if I plot the particles here to scale, and we go from a 10 micron particle to a 1 micron and even smaller, you can see the 1 nanometer particle still. It is about 1 pixel at this resolution of my image. But one important thing to keep in mind is if we go from larger particles like these to the nanometer size range, and we keep the volume constant, every time we go 10 times smaller, we have 1,000 times more particles. So if we assume that we really fragment a 10 micron particle into smaller fractions, we will end up with a very large number of very small particles. So just to give you a number, if I fraction one of those 10 micron diameter particles, I will get 1 to the 11 particles with a 2 nanometer diameter. And just to link back to Lucy counting stars, we estimate that the universe contains about the same number of galaxies. And just to link maybe also to data analysis, if we want to list just the size of this number of particles with an integer number, we will need about 500 gigabytes of data storage. Now, I brought this example because now the first thing you will say is, oh, that's not very realistic what you're telling me here. But this is exactly what we are interested in, looking at this physical chemistry point of view. So how are these nanoparticles actually forming? Are they directly coming from fragmentation of larger particles, or are they directly forming from very large bulk material? Do they gradually become smaller, or do they form directly? How do they form? What's the chemical principle behind it? How fast do they form? And specifically under what conditions? So especially if we look into impact and interaction with the environment, but also with humans, then we need to consider conditions that are different from just atmospheric conditions. And at what size do they actually disappear? And I'm not talking about the resolution limit we have with our methods, but at what size should we not talk about a nanoparticle anymore, but maybe of a macromolecule, which is in a similar size range when we go to very small particles. Related to that, also, what densities do we actually have with these particles, and what properties, specifically surface properties, do they have in general? Because these are, of course, important when they interact with the host. So much of this we know for metallic nanoparticles, but not for nanoplastics. And of course, the biggest question that remains is how do we visualize them? So one method that we use in our lab and in the consortium is force microscopy, and specifically combined with infrared spectroscopy. So in that method, very, very briefly, we use an AFM tip, as you see in the top left, to raster scan the sample with a very, very small tip. Let me see if I can get the laser pointer here. With a very, very small tip, we raster scan the sample. We get a topography image of any particles that are on a flat substrate. And at the same time, we shine a laser light focused just below the tip on a specific spot. This laser light induces a dipole change in our sample. The dipole is mirrored in our tip. And this dipole-dipole interaction causes a force that we can measure. And from that force, from this interaction, we can reproduce a spectrum that has the characteristic infrared bands that we can use to do infrared spectroscopy and identify, for instance, specific plastic species. So what do we get from this method? We get a topography image, so we can see sizes, we can measure their height, but we also get spectroscopic information. As you can see here, for instance, we can map at a specific infrared band that might be characteristic for a specific species of a specific band vibration that tells us something about the species and the chemistry of the sample. We are in a resolution regime of down to 10 nanometers and slightly below. We have a very shallow probing depth, which might be interesting to investigate, for instance, surface chemistry. We can do this hyperspectral imaging. We have a very limited field of view. Obviously, it's always the compromise we have to make. If we go to very high resolution, our field of view will be limited, and that also limits, of course, the number of particles or number of stars we can actually count. So we get particle numbers, but this is, of course, limited by the time we need to inspect our field of view. We get particle size, particle shape, and we get an infrared spectrum. And here I show you an example of something we've published last year, where we looked at the omnipresent polystyrene spheres that are used a lot in research at the moment. These have a ligand to stabilize them, to prevent them from clogging together. As you can see here, these are the particles as purchased. And what we saw here, what we see here, if you look at the spectrum we recorded for this individual nanoplastics here, we can detect the sulfonate ligand of the stabilizing ligand around the plastic particle in the SO double bond stretch vibration here. And after we treat this particle with hydrogen peroxide, we were able to remove this ligand here. And what we get then after this removal here, a spectrum recorded for an individual PS sphere here, we get a spectrum that very much resembles what we would expect from a PS bulk, just as a reference here, recorded with bulk infrared spectroscopy. So just as an example of what we can do here with this method. Now, the next question you might ask is, okay, how does that help me with my samples? Well, there are a couple of restrictions, and this is now I'm coming to the actual challenges that we face when we look at nanoplastics from the environment. We need to deposit them onto a substrate. So in the simplest case, we can just apply or pipette a nanoparticle suspension onto a substrate. Often we use spin coating, trying to achieve a homogeneous deposition of these particles on the substrate. Then we dry it, hopefully under conditions that do not alter our nanoplastics, and then we can measure them with our AFM. So far, so good. But this is, of course, not always so easy if we have more complex samples. And one thing that we are at the moment investigating is using suspensions and filter them. And this is an example of a filter system that was developed in the Polyrisk consortium, where we have, for instance, a two-stage filtering to remove larger particles that are going to interfere with our high-resolution scanning. And then we have a second filter here with a specific pore diameter that captures all the particles in the size fraction between the pore size above and the pore size below. And then what we can do is we can directly look at this filter membrane here, for instance, an aluminum oxide filter, but also what we tested are gold-coated polycarbonate filters with various pore sizes. And then we can look at the nanoparticles deposited directly on those filters. Why do I highlight this specifically? In Polyrisk, we also have an aerosol project, and these are filters that might be useful for direct, or that are already used for direct aerosol collection onto these filters with impactors to collect aerosols. And then we can look at those filters either directly or, and this is what we have to test now, after some pre-processing to remove a very large particulate matter that might end up on the filter as well. But this is ongoing research here. But these are the approaches we have identified so far. Now, if we go to more complex sample matrices like blood or serum or human tissue, this is not going to be successful. We are going to need additional steps that include pre-processing, digestion, additional filtration of our samples, probably also pre-concentration step, so that we actually have the chance to find these particles after we apply them onto our sample carrier. And in order to optimize this procedure, what we are planning to do is that we combine this pre-concentration with a filtration step that will be able to segment our size fractions into different batches. And then we can, with, for instance, mass-based methods, try to detect a certain mass that we say, okay, there is a certain mass distribution as a function of size. And based on the mass that we detect, we can then decide that we have a relatively high chance that if we use a certain size fraction with a minimum mass, where we found the minimum mass inside, that we have actually a chance to find individual particles when we apply, for instance, 1 or 10 microliters of that batch onto our sample carrier. Because otherwise, we are literally tapping in the dark. If the mass concentration is too small in these batches, we will have statistically basically no chance to find individual particles if we apply them onto our flat substrate. So what we are tackling with this approach, or trying to tackle here, is the problem of low concentrations and the statistical significance. Because as I said, here we can count tens, hundreds, maybe thousands of particles, but we will never be able to count the number of galaxies in the universe. So where are we with this? At the moment, we are working on digestion protocols, on filtration and separation approaches, and on pre-concentration methods to implement what I've just shown you on the previous slides. We have tested, we are testing mainly with commercial polystyrene spheres, but here we would, of course, be very interested to obtain also other particles, as most partners, I think, in this project at the moment, to get something else than the commercial particles. We are testing this at the moment with water, in water solution, but also already with cell cultures that have been spiked. And also we started working with tissue in the first experiments. First results I can report is, yes, we can find the micro nanoplastics back after our spiking experiments. We test this with confocal fluorescence microscopy because these are still fluorescent particles, and also with AFM. We have tested a different digestion and exposure, different chemicals that we use to digest. And what we see is that different treatments cause different maybe problems, like particle agglomeration. But in principle, we are positive that our digestion protocols work efficiently without altering the particles, the plastics that are in the matter that we want to digest. And we've also tested extensively now the gold polycarbonate filters, because they also need at some point to be resistant to our chemical treatment, and that is of course important for the filtration step. So ongoing work, as I mentioned, we are looking at spiked samples. Here an example of blood serum. We are looking at self-made PET particles, where we just do a very simple mechanical grinding and see what we can find back. We are looking at the commercial polystyrene spheres to optimize our analytical methods. We also look at samples from the environment. We already found some nanoplastics in ocean water, but also we focus on degradation studies, where we try to understand better how nanoplastics form. And we also have some ideas about capturing particles in patterned substrates, where we use little sinks that might attract particles, which will help us a lot to maneuver and to orient ourselves at this high resolution on the substrate that we are studying. And with this, I hope that we will have something better than just the chair Lucy is using to improve her analytical method. And thank you for the attention. Any questions?
Okay, so we have a question from Anna Costa in our chat. I don't know if you can see that, Florian.
Wait a second. Yes, I should be able to. Yeah. Can you see the chat now as well? And everyone can read it? No?
Okay, I'll read it out loud if that helps.
No, I can see it. I can see it.
Okay.
For some reason, I cannot move my chat window. Wait a second. Maybe I can. Now I have it. Okay. Good. So, first question. Anna Costa. Can everyone, should I read the question or can everyone see it?
Yeah, because I can't see it on your screen. So perhaps read it.
Can you see it now?
I don't think you can. I'm not too sure that you can share the chat.
So Anna is asking, how can we be sure that the digestion protocols don't dissolve degraded plastics? So, well, that's exactly the point, right? So what we do at the moment is we spike samples, we treat them with the chemicals or the enzymes that we have, and then we try to find them back. And we know what we put in, and then we know what we should get out. And with the PIFM, for instance, we can check if there are morphological changes, and we can also check if there are spectroscopic changes that tell us that the chemistry changed. Does that answer the question?
Yeah, perfectly. Thank you.
Thanks.
Thank you very much.
Jane is asking if she can know more about the bottled water work. Well, yes, I can comment on that. I can talk a lot about it. So what we do is we take PET bottles and we store, or we take the water out of them, and we do our pre-concentration and filtration steps, and then we see if we find any nanoplastics back. And what I can tell you so far, and I don't want to say more, is that we can find some. But this is, I know that Corinna Altmann, I think she's also here. I think they have even more detailed results. And I think what was shown in her work and previous work is that specifically the opening and closing of the bottle produces micro nanoplastics in the water, because seemingly due to abrasion of the cap, there's an increased concentration. We are not focusing on that because that was already shown, but really just on whether we can find micro nanoplastics or nanoplastics back in the water that is for a certain amount of time in these bottles. So we just buy them in the supermarket and we try to find something in there. But I can tell you more if you want to see these results. Anita has a question. Regarding the blood samples, have you encountered any background interference from plasma, and how are you overcoming it? Also, have you noticed any bioattachments on plastics? That I cannot answer yet because we are not yet there. So we are in the process at the moment of trying to digest blood samples. So I don't know yet to what level we have to deal with interferences. I expect that we have to do something there. We have Adrian and Laura both working on digestion protocols for exactly that reason. And then we will have to see what problems, what other challenges we face there. So I cannot answer the other question about bioattachments, because so far we have not seen that. What I can tell you from the ocean samples is that almost always we find nanoplastics attached to some other probably biologic material.
Okay. Thank you, Florian. I'll have to stop you there. Well, firstly, I know you've got to go to another conference, and also because of the time constraints. Thank you so much for your presentation and your input. We've got one more question here. Yeah, we've got time for this. Just a quick
Yes, yeah. Ideal substrate for BIFM. So what we use at the moment, it has to be very flat. That's the main requirement. So what we use at the moment is commercial silicon wafers, but we can also use gold-coated polycarbonate filters. They're flat enough, and the gold is actually beneficial for the method. So all the filters, aluminum oxide, silicon wafers, or these gold-coated polycarbonate filters, as long as they're flat enough, and I'm talking, well, you see that we have a few nanometers precision in height measurements. As long as they're flat enough, we can use it. Thanks. Okay. Thanks, everyone.
Great. Thank you, Florian. Right, I'll hand over to Mahdi now.
Sure. The next speaker of our workshop is Dr. Tao Huan. Dr. Huan is an assistant professor in the Department of Chemistry at UBC. The focus of his presentation is going to be on the applications of mass spectrometry-based metabolomics in studying microplastics. Tao, please.
Hey, hello, everyone, and good morning and good evening. It's actually really early for me, although I actually, in about 15 minutes, I need to teach my course, Chem 211, Analytical Chemistry. So I have to leave early, but I'll try to finish my presentation first. So today I'm going to, I'm an analytical chemist. So we develop a mass spectrometry-based method to do metabolomics. To begin with, let me turn on. So to begin with, metabolomics is a study of.
Sorry for stop. Would you mind putting it in the presentation mode?
I think it is in the presentation mode.
Is it? Oh, okay. So maybe it's with the way we are seeing it. It's with, okay. Sorry.
How about now? Is it in presentation mode?
Yes. Perfect. Thank you.
Okay. I think there's probably some delay here. Yeah, so the metabolomics is study of all the metabolites in a given biological system, including cells, tissues, or biological fluids. Metabolites are the downstream products of gene and protein activities. by studying the metabolites, allowed us to have a better understanding of health and disease, have a better mechanistic understanding, as well as identifying biomarkers for disease diagnosis and prognosis. And a typical metabolomics workflow starts from sample preparation. We first extract the metabolites from these biological samples, and then we perform a liquid chromatography mass spectrometry-based analysis. And further to that, because it's generally a large amount of data, we need to do the data processing to extract the metabolites from the raw LC-MS data. And from there, we do statistical analysis to identify those statistically significant features. Our understanding is that if anything is statistically significant, it infers biological significance or biological importance. From there, we identify these metabolites and then trying to do biological interpretation. So this is what we do in the lab. We trying to develop a better analytical method and better bioinformatics to identify, to understand their biological meanings. How about, oh, sorry, I think something wrong with the Zoom. It crashed. Can everybody hear me now?
Yes.
Okay. Yeah, sorry, somehow the Zoom crash never happened in the past 3 years, but just, yeah, so that's what we do. And in particular, in terms of application side, we are interested in applying metabolomics to different biological research questions. So one thing we're interested, exposome, which is the totality of environmental exposure that the individual could experience. The reason why it's so important is because for the disease that we have, in many cases, it's genetic risk, but also it has some environmental risk as well. So that brings me to get into the exposome field by using metabolomics to quantify the entire set of chemicals that's available in our environment. And another thing actually being interesting for a very long time is how the microplastics, because these things can actually get into the marine organism, then transfer through food chain, and they can accumulate and cause some biological damage. Yes.
So I'm very sorry to interrupt you again. I think something is, so your slide is just on the research focus and human lab, and we cannot see it. I think you're moving forward, but now it's this next one, metabolism. Sorry.
Okay, really sorry about that. I have no idea why this happens. Yeah. How about, is that now in the metabolomics in exposome?
Yes.
Okay.
It's not the presentation more, but it's okay. Don't worry. Yeah.
Sorry about that. Yeah. I should probably restart the computer before I start. Anyway, so yeah, so this is the exposome, and we are interested in the microplastics. And we believe that this is very, it's a very global environmental issue, and it's a research hotspot. So what we can do from the metabolomics perspective is to understand how microplastics, trigger the biological consequence, how it causes the biological response, and how it have a toxicology mechanism. So for last week, I got contact to give a talk there. So maybe I really don't have anything research work ongoing related to microplastic. Then I look at my publication list. I did, in some way I collaborate with people, and we did a toxicology study of how the polystyrene microplastic affect the marine mussels. So I think it'll be probably be a good idea just to present this work. Okay, can you see my experimental design slide?
No, it's not. It's not moving. It's just it has stopped at the one that you brought at the beginning. Now we can see it. Yeah. So
okay. How about this? I'll just. You have no idea.
Do you?
Is it now? Is it an experimental design slide?
Yes. So just when you're maybe moving to the next one, you can double check with us because.
Okay, sure.
Yeah.
Okay, so the way they did it is they study muscles, and then they basically have this 30-liter big glass tank, and they can take a look. They have 4 different plastic concentrations with 6 replicates per each. So that being said, they're gonna have a 24 these 30-liter tanks. It's a big tanks. They have 24 replicates there, and for each tank they have 15 muscles. They basically put the 15 muscle there, and then add micropolystyrene. And then the concentration of micropolystyrene is basically follow the literature report. So they have 10 particles per liter, and that is environmental concentration reported in Yangtze River. And then they also have 10,000 particles per liter, and that is a recent report from Pearl River. And then or even higher, that's really a pollutant, concentrated 10 to the 6 particles per liter. And also, of course, they have a negative control using clean filtered seawater, because the mussels are growing in seawater. So these are the 4 different microplastic conditions. Then what they did is they put mussels, add the seawater, and put the polystyrene there, and then do 14 days of exposure. And after 14 days, they will transfer all the mussels to another 24 new glass tanks for a 7-day recovery, because we think they've been suffered through these high concentration plastics, and so they need some time to recover. So can you see that now the experiment design 2? Okay, perfect. So this is how the schematic workflow. Day 0 to day 14, microplastic exposure, and day 14 to day 21, recovery. And then after day 7, we collect sample. Day 14, we collect sample, and day 21, we collect sample. And these samples represent, they suffered through the PS, the exposure. Then 14 days, even more. Then 21 day, they got recovered. Then we do the, extract these biological tissue samples and do metabolomics and also biochemical measurement. Now I'm in the experimental design 3. So it talks about first we dissect digestive trait and then do fluorescence analysis using the microplate reader. And then we also extract some biofluid for biochemical measurement. So I'll talk about more how they measure these radical organic, these ROS and other organic radical stress. And then for metabolomics work as well. So first, let's take a look how the micro PS accumulation and the clearance. So first, they noticed that in the control, the negative control, also 10 particle per liter, there is no micro PS accumulation. So basically, they do fluorescence analysis, at least probably related to their detection limit, but at least they couldn't find anything there. And then for these more higher concentration groups, 10 to the 4 particles per liter and 10 to 6 particle liter, they can actually detect the micro PS that's being accumulated in day 7. And then you compare to day 14, you can see got a lot more concentration, right? And day 21, because they're being recovered, the concentration of the microplastics decreased, but it's still not completely cleaned out. It's very interesting. And then if you do 10 to 6 particles per liter, you will see that it goes nearly. So this is a unit, 10 to the negative 3 microgram per milligram. So with that, you can say that it's about 0.9 microgram per milligram muscle tissue. And then after 21, it decreased to much lower value, but it's still there. I'm not an environmental chemist, but I think this shows that it increases after you put it in the microplastic environment. Then if you take it out, the concentration decreases. And then they also studied oxidative stress and the immune response, using reactive oxygen species in ROS and all these different. I think these are the biomarkers. I'm not in that particular field, but they can actually see. You have a control 10 particle. 10,000 and 10 to the 6 particles per liter. You can see that how the these RS level goes up. Day 7, day 14, day 21, and with higher concentration, it will of course have a a higher oxidative stress. And so it's very clearly show that after the muscle being exposed to this microplastic environment, lots of biomarkers have bigger changes. And further to that, we did a metabolomics work. So we took 60 microliter, it's a very small amount of muscle hemoglobin, and then we use methadone and water to extract metabolites from there. After we extract the metabolite, we perform the LC-MS-based analysis to collect metabolomics data. So on the left side, that's the LC-MS system, and we use a LC column to separate the metabolites based on their different interactions with the stationary phase. And then from the LC chromatogram, it actually contains a lot of data. If you click any point, you will get into the MS spectra and they show you all the, each peak will represent one metabolic feature information, and we can get at over 100 features per spectra. And in one LC-MS run, there are thousands of spectra there. So that's give you quite a large number of metabolic features. So from there, we use data processing software to process it. In total, then we can confirm over 3,000 metabolites in the 24 samples. And then once we got that bigger data, we have thousands of metabolites and we have many samples. The first thing we can do is do principal component analysis to gain an overview of how these metabolites are being changed. So each spot here represents one sample, and a PC plot showing you how close these samples are related. If two spots are close to each other, means their metabolites are more similar to each other. If they are far away, if the yellow dot and the green dot, if they are far away from each other, means that these two samples have a big metabolic difference. So we have control in blue, control in green, and then you have 10,000 and 10 to the 6. You can see if you compare day 7 to day 14, you can see how these control start to separate from these individual samples. This indicates that after 14 days exposure to microplastic, the overall metabolic levels, the metabolites have seen bigger changes. So that's how they start to spread away from each other. But then you take a recovery for 7 days, you will see how these things will come back together again. So day 7 you have a pattern, and then you spread, have a better separation, but then you get closer together because now these have enough recovery, and then their metabolic level back to be more similar to each other. So principal component analysis allows to give a overview of the metabolic changes. And then we can also dig into the data to figure out which metabolites are really being changed. So for example, in the study, we find 5 different metabolites being have a significant concentration difference in different days, as well as in different treatment groups. And these metabolites can be used potentially as biomarker to tell the pollution of microplastics. On the other side, it can also be used to have a better mechanistic understanding of the microplastic pollution. So one thing we can do is do a correlation analysis. For example, we have all these biomarkers, right, related to oxidative stress and immune response, and we can use these. We can do correlation analysis to correlate those biomarkers with this metabolite that we discovered in our metabolomics workflow. And then we can do correlation analysis where a darker color means it has a more stronger correlation, with a blue color means it's negatively correlated. So our result shows that there is the biochemical indicator and the different metabolite. There's certain level of correlation, which suggests that, for example, probably the ROS is correlated to some of these metabolites. And further to that, we can also perform a pathway enrichment analysis to map all these changed metabolites into these human endogenous or these muscle endogenous pathways to understand how these metabolites are related to each other or relate to oxidative stress and others. Okay. So I think because of time limit, that's it for my presentation. Sorry for the problem with the Zoom, and I'll be happy to answer your questions.
Thanks very much, Tao. Are there any questions for Tao? Please, you can speak, mute your mic, or you can put it in the chat. Okay, no, can't see. Okay, so it looks like, oh, Maite has it. Please, Maite.
Hi, Tao, thank you so much for the presentation. So I have a question. If you were going to use another organism beside mussels, what is the ideal sample to collect? Like, can you collect tissue, or it has to be liquid, or it has to be what do you think is the best?
Right. So I think it doesn't matter. From a metabolomics perspective, we can do metabolomics to tissue, to biofluid, even to the shell. You know, we can do metabolomics to any biological sample. But I think the more important question is what will be the thing that get most affected by this microplastic pollution? So that will be, you know, we can, you know, dissect, and we can cut the tissue, and then we can do you know, tissue of different position, different locations, and then we can do, you know, spatialize the metabolomics. So that will probably have a better, because I don't know which part will be affected the most, but we can, because we only need a very small amount in terms of tissue. We only need about 10 microgram. 10 milligram in terms of biofluids. We only need, you know, 50 or 100 microliters. So it's a small volume. So we can actually, you know, do these different parts of this muscle tissue, and then see which part being affected most.
Okay, thank you so much.
Yeah.
Thanks very much, Tom. So I think, Lesley, I will pass it to you. I think this looks like, yeah, there's no more question. Thanks.
Great. Thank you, Tal. Thank you for the interesting presentation, very in-depth. As you know, there will be slides of everybody's research put onto Zenodo. So once again, feel free to submit them. I'm now going to hand over to Anna Costa. Anna is going to, if I get this right, present on strategies for NP simulated samples and laboratory tests. And Anna is part of Working Group 1 and PlasticsFatE, and coming to us from Italy.
Okay, thank you very much for this kind introduction. Do you see my screen? Not in the presentation mode, I suppose. Now is right?
Yes, we can
okay,
see.
Perfect. Thanks. Okay, good morning or good afternoon, depends everybody. My name is Anna Costa. Yes, as Lesley said, I comes from, I'm here on behalf of PlasticsFatE project, Work Package 1. I come from an institute of CNR in Italy, Institute of Science and Technology for Ceramics. So actually my background and field of expertise is on inorganic materials. So I'm here and represented the work package for the characterization, physical chemical characterization actually of plastics in PlasticsFatE for the experience that I gain with nanomaterial but of inorganic composition. So some things are also new for me, so I'm very happy also to participate to this workshop to learn more about this new nanomaterial. Okay, nano or macro material. Okay, so in this slide, I briefly show the activities that are addressed so far, but also in the future for Work Package 1 in PlasticsFatE with sample in simulated exposure conditions in order to identify, extract, characterize, so optimize method for the characterization that can be suitable when the sample are contained in, when plastics are contained in real sample. So this activity regards the preparation of plastics, the selection and preparation of plastics through milling treatment, aging, weathering, to obtain what is the PlasticsFatE portfolio nanoplastics. The basic characterization of such plastics in order to define their physical chemical identity and report it in a sort of technical data sheet so that plastics can be used also by other partners interested to this investigation. The development and validation of protocols for sample dispersibility. This is an issue that we were also discussed at CUSP level. It is something that is a key step for all the method developers or all the people that characterize plastics, and also method for the extraction and characterization of plastics in such simulated metrics that for the project are some environmental metrics, food, drinks, inhalation air and personal care products, and also human tissues and fluids such as lung, gastrointestinal tract, lymph nodes and blood. Okay, so as a first action, we selected some representative materials that should that differing for the polymers type, the source size, the use scenarios, and the presence or not of significant additives. So this is more or less the rationale, and this is the code that we introduce to identify in a unique way the samples that, of course, can be shared at CUSP level if it is of interesting for the other projects. In this slide is shown the PlasticsFatE portfolio that contain a list of samples that were prepared by BAM, optimizing procedure of milling, weathering, characterization for checking batch-to-batch reproducibility, and recently also for check endotoxin contamination. In addition, we have a sample that was provided by Clariant called Ceridust, that is of lower size than the sample prepared by us, so around 5 microns. We also are in touch with Plastics Europe in order to see if some other commercial sample can be considered, and we also purchase a nano emulsion of polystyrene doped with lanthanum and europium for labeling purposes. Well, as a second issues, we are preparing such a technical data sheet, as I said before, reporting the physical-chemical identity of sample. Fraunhofer is in charge of this activity and has selected some basic physical-chemical properties that can be useful to identify sample and to predict to predict their behavior when they will be dispersed in complex metrics, or will be present in complex metrics. Here is reported some images that will be part also of this quite a huge technical data sheet. This is just an example in order to made by BAM in order to check if the morphology of particles change when passed from the powder to the powder suspended in a water medium. And what they notice is that the initially round the edge shape powder becomes with this irregular shape in suspension. The third issue, very important as told before, regards the dispersibility study. The dispersibility issues, yes, is something that is of primary importance also for the other project. So we had an internal discussion that, of course, is open at CUSP level, and agree to adopt a two-steps approach inspired to the well-known nanogenotox protocol used for inorganic nanomaterial, that consists in the preparation of a general dispersion that is called stock dispersion, that should provide the as much as possible stable dispersion, And then this stock dispersion is used to further dilute the stock in the exposure conditions in order to obtain such work dispersion, and this can facilitate reproducibility within different tests and operators. Here are reported, I will talk later, but the dispersants that the surfactants that we choose for preparing the stock dispersion, we compare different surfactant, different concentration of powder, different concentration of surfactant. We tested the suitability of ethanol treatment, of milling treatment, in order to provide a protocol that was the best one for these purposes. And also here, the University of Turin has already started to test, to prepare the work dispersion with surfactants that are close to the exposure conditions, so to the study of inhalation, or ingestion, or blood transfer. Here are reported some results. In order to prepare the stock, we tested 3 dispersants. The well-known BSA, what's suggested by nanogenotox protocol, the twin that is a surfactant that was suggested by polymerists because suitable for dispersing polymers, and is new entry, a biosurfactant that is known for its very good performance in terms of dispersibility of colloids. We perform some floating stability test in order to check the time that in this case, we tested the cherry dust high density polyethylene of 5 microns, and we compared a different concentration of this biosurfactant and at quite high concentration of the other 2 surfactant, the floating kinetics. Then we took the best surfactant that was considered sodium surfactant, even if now we decided to go, in any case, to go ahead with BSA that is well known and no concern about toxicity or rise. But in any case, we tested all the portfolio material of PlasticsFatE, and we noticed that in some cases the dispersion were good and stable. In other cases, when the particles are too large, of course they separate quite immediately in the presence of surfactant, and the particles with higher density than water, of course, sediment, the other one float on the surface. These dispersion were then observed at the optical microscope because particles are quite large, and in this way we could also analyze and extract a particle size distribution that was coherent with what provided by BAM when they produce such particles. So the conclusion is that particles does not aggregate, so they don't need surfactant for breaking the aggregates, but need surfactant in order to have a to avoid and to slow the process of separation from the water. Here are the preliminary tests made with in the exposure conditions. So with, in this case, with bile used as surfactant, and again we could show that bile is an efficacious dispersant for both polyethylene and polyethylene. Nevertheless, the sample sediment or float over time for the reason said before. In any case, the good thing is that also for stock dispersion, but also for working dispersion, they can be, in most cases, they can be easily redispersed by a gentle mixing. Here with UVV spectrum you can see the sedimentation kinetics over time. In this slide, as far as the extraction methods are considered in such simulated exposure conditions, University of Bayreuth, in collaboration with Innosive Diagnostic, tested such filters that are commonly used to analyze microbes in order to collect sample and to see if sample collected were suitable for microFTIR and Raman spectroscopy, and the preliminary results was quite good. Finally, this is an example of characterization in real exposure conditions, an example of validation of the method of characterization in real exposure conditions. What University of Bayreuth did was to incubate 6 different polymers in tissue fixatives that are commonly used for toxicity test, and they compare different plastics, not from the portfolio, but plastics that were already present in the laboratory, and they after did a digestion protocol, as mentioned before by Florian, and what they do was to compare the Raman spectrum before and after this treatment, and apart from PLA that completely disappear after the digestion, so is digested by the hydroxide, all the other samples show Raman spectra that was comparable with the sample before the treatment. I just finished such a brief overview of the work ongoing in package 1 in PlasticsFatE, and I would like to thank again CUSP for this kind of invitation, and also for organizing all these meetings that are fundamental, in my view, to share and compare results and agree common strategies for the future. Thank you very much for your attention.
Great, thank you, Anna. That was a really good overview and hopefully of interest to everybody here. So please follow up with Anna if you have any questions. Oh, we've got one here from Raymond asking if you can tell more about the biosurfactant.
Yes, this biosurfactant actually, yes, I called it a new entry because I was testing for other reasons, because such biosurfactant is commonly employed in cosmetics matrix because it has really very, very high performance in terms of producing microemulsion at lower concentration, 10-fold lower concentration than the other surfactants commonly used, and this was found also for plastics. It works very well, but the concern, at least in our consortium, is that this biosurfactant it is a lipopolysaccharide that comes from the bacteria fermentation.
Oh, really?
So, yeah, so the first concern arises if some of this bacteria contamination could remain, and our partner for toxicological studies is STAMI partner, is testing for the possible contamination or the possible residual of material that can be toxic itself. What is reported in literature is that, yes, it is bioactive. It is also known for being antiviral and antibacterial substance, but the cytotoxicity limit is quite low. At least we used at lower concentrations than the cytotoxicity limit reported in literature. But of course, all these things should be verified. And in any case, our consortium, since the beginning, prefer to not go into new possible complication, but to prefer.
Yeah, well, if it's really LPS, that's what you said, lipopolysaccharide.
Yes, a lipopolysaccharide. It has.
Yeah, that's very active on macrophages, yes, as you know. So you will find immune effects if you treat them like this. That's what we find.
I don't know if it can become a model maybe for investigating some. No,
It's a positive control.
Okay, positive control. Okay, great.
Great, thank you, Anna. Okay, unfortunately, we don't have time for more questions. I'll have to hand over to Mahdi to introduce our final speaker.
Sure. The next speaker is Dr. Ed Grant, the professor of chemistry at UBC. Ed is supporting the characterization and quantification of a number of projects here at UBC, and is going to share some of the insights that he has gained, and his team has gained through these ongoing collaborations. Ed, please go ahead.
Thank you, Mahdi. Let me bring my screen up here. Yes. Good. So let me begin by saying I'm very happy to have found myself a member of this cluster at UBC. It's been a very interactive group, a very stimulating group, a lot of great discussions, and I think some very interesting work has come out of this. And I'm also especially excited by the prospect of forming a link with European partners. I think this is a very big problem, and it benefits from having so many ideas and so much input. And also I think that the geographical points of view can play a very important role in coming up with the most creative solutions. So let me just say a word about my effort. I'm in the Department of Chemistry at UBC, and about half my program is focused on the analysis and classification of complex materials. And for us, anything interesting is a complex material. Some time ago, when I was in the US at Purdue University, I formed a company that built portable instruments for many kinds of analysis, but one of the things that we were good at is the analysis of post-consumer plastic waste. And so we were involved in certain kinds of recycling projects, and for a time, our little company was the gold standard in plastic materials identification. Well, that work I kind of left that behind, but then the cluster found me, and I had the very good fortune to get involved in a collaboration that was spearheaded by Brian Hunt with his student, Natalie Meirer. And my student, Matt Kowal, got involved in this, and we analyzed some organisms from Baines Sound for microplastics composition or content. And this really produced some nice results. Let me get myself able to advance the slide. Yeah, there we go. So we used, Natalie did all of the work in capturing the organisms, mostly plankton and some herring, and dissected them and extracted plastic samples visually. And we used our Raman microscope system to analyze the products that she found. And we found, I mean, it worked just as well on very small particles as it did in our old plastic recycling research. And we used all of the methods of spectral processing and machine learning to develop libraries and analyze plastics to those libraries. And it was a very successful effort. I think we managed to characterize what the profile of plastics were in this particular fishery. But it led me to start thinking about really a problem in the challenge of being truly representative in any kind of microplastic analysis. You know, going all the way from the environment to the laboratory, how do you know what you're looking at really reflects the distribution of materials, and especially the level of concentration. How effective can you be in answering really important questions having to do with what's out there in the environment based on what you see in the laboratory. And this challenge of representative plastics, microplastics analysis faces a variety of challenges. There's material challenges that come from a very broad size range that one must deal with. If you're going to address microplastics in the environment, you should be thinking about sizes from nanometers to millimeters. There can be quite a variety of polymer substances, and we've seen examples of that today. Aspect ratios, well, the most dramatic difference is probably a very common one, maybe even a 50-50 difference between fibers from clothing, dryer vents and so on, and wash water, to spheres that break down from solid materials. And one also has to reckon with the additives and contaminants, that is plastics and their ability to absorb pollutants and support biofilms. And there could be quite a wide variation in the state of aging. So these are all challenges in trying to form an assessment or a global picture of a particular environmental system and say what's going on based on observations in the laboratory. And then in sampling, in microplastic analysis, you confront microplastics in air, water, and soil. And the matrix can be quite important. Solid waste, a landfill, sewage, in organism digestive systems, and so on. So there's really an issue of sampling and how can you be representative in what you're taking from the environment and then projecting back your knowledge into what that really constitutes as a load. There's also the question of dilution. We live in a world of natural particles, dust particles, soot, microorganisms in the air, vesicles, aerosols. You really, our business is to distinguish between these natural sources that our bodies have developed an ability to live with from microplastics and try and develop some assessment of what the unique effects of these distinctive materials are. Also in sampling, one has to worry about recovery efficiency and the possibility of transfer losses and even contamination from ambient sources in a laboratory. So these all present some significant challenges, I think, and things that we're worried about, at least I'm worried about in my lab, in trying to connect back the observations that we see. In general, when you're out in nature and sampling, your goal is to separate and extract microplastics from the environment, and people use, as we've seen today, screens, sieves, filters, size exclusion, questions of fouling. There are density separation methodologies, but one has to be concerned about losses and density fractionation. People apply digestion methods, strong oxidation, and other means of getting rid of biogenic materials. And there's some chemistry issues here. There have been some clever methods applied to extract plastics based on their hydrophobicity using oil and absorption separation. These are introduced steps of materials processing where you really have to worry about the quantitative assessment of that. And something that really should be done routinely, and you don't often see this, are methods of standard addition, where a person who's undertaking a study of the environment identifies a particular kind of plastic of interest and does studies very systematically where you add known amounts and make sure that your recovery reflects a perfectly acceptable extrapolation back to the native sample. So this is kind of a summary of things that I've seen in recent reviews that validation is really a very, very important consideration in as important as really clever analytical techniques. One has to worry about validated methods of sample acquisition, separation, and analysis. And this also brings us to another question, another, maybe a very small elephant in the room having to do with nanoplastics. And these tend to escape our notice because they're so small. but these may be the most impactful plastics of all. And there's a Nature article that just came out that calls attention to that. And I took a table from Lim's Nature article to indicate the publication pressure as a function of particle size. And I added those gray columns to indicate that under 1 micron, there's almost nothing published, and really nothing very much in the natural environment. And this has been an area that we've been attracted to. Certainly, when we're doing Raman analysis and micro Raman, it's quite simple to, once you've got a sample and you're satisfied with the acquisition conditions and you can see the plastics in the microscope, it's actually a very straightforward problem in a variety of ways. Raman is one of them, to go in and get a sample identification. A real challenge comes when you're dealing with plastics that are too small to see. And this is an area that we've been developing in connection with other interests in nanocrystalline chemistry and in protein chemistry. So we've developed a microscope that's able to use principles of interference to see beyond the diffraction limit. And we found some ways that we think are distinctive for recognizing and automatically counting microplastics in a field of view where we're focused on very small particles. And let me just conclude by showing you an example from an experiment that Matt Kowal has done. So this is uses a methodology that we published recently in connection with gold nanoparticles. So this is a cover of just late last year in Journal of Physical Chemistry, where we are showing here a methodology for resolving individual gold nanoparticles. And the arrow is pointing to a Gaussian fit to a video, and we can watch these videos as nanoparticles too small to be seen by optical microscopy create an interference signal that we can relate to particle size. And so we can do particle size, and with certain kinds of selectivity in cover slip engineering, we can do a kind of surface chromatography that identifies plastics. And let me show you a movie of a real experiment. This is streaming. This is what we see in the laboratory when we're doing an experiment like this. And I'll show you the landing of 50 nanometer polystyrene beads on a cover slip, just taken from a solution. So this is a solution you micropipette from a sample you might have recovered, put this in the well of our microscope, and watch the landing of these sub-diffraction limited microplastic particles. And this is what the experiment looks like. There we go. So to go with this kind of data acquisition, we've developed automatic particle tracking and counting software. So it's a 1-minute job to count a sample with 4,000 microplastic landings. That's a very routine measurement for us. Oh, this is a raw video. So what you saw there was somebody putting their hand on the table, and this is something you can barely see in the image before all of this processing, but it makes a pretty dramatic effect here. So this is essentially a hands-off, manipulation-free, direct visualization of microplastics. Now, here you're seeing a fairly uniform size distribution because this is a commercial sample that's advertised to be 50 nanometer diameter particles. This methodology applies with very little modification in concept or practice up to 100, 200 micron size particles. Bigger than that, we would use conventional microscopy to see. But as long as we're dealing with nanoscale materials, this is the sort of methodology that we prefer to use. And so I'll just leave it there. I'll be brief and keep us on schedule, and happy to answer any questions.
Great. Thanks very much, Ed. Any questions for Ed? Yeah, so feel free. I think we're out of time, so feel free to email him. And I think the publication that he showed has showed at the beginning is still in press, so that will also be available very soon. But this is also ongoing. It is. Oh, it's online now. Perfect. Okay, great.
Yeah. So if you want to look at this methodology applied to gold nanoparticles,
the nanoparticles one, yes, for sure. Yeah, perfect. So okay, Lesley, I think pass it to you to end.
Yeah, we're out of time, so we won't have time for the Mentimeter nor time for the individual slides. What I will do, though, is put them on Zenodo along with a recording of this workshop. And I will send the link to Mahdi and to the contacts that I have so far. So hopefully you'll all be able to find it. Thank you, everybody, for attending, because I know it's very early and quite late. But yeah, it's just been great to see the participation and the engagement of everybody, and particularly to the speakers. And a thanks to Michelle Epstein. I don't know whether she's here or not, because this was her idea. The topic area was her idea in the first place. And yeah, thank you everybody for your support in sharing this event. And hopefully we'll have another one soon. I think there's one about mouse lungs coming up, but we'll let you know anyway.
Lesley, there is a question about where people can find the recorded session, the recording.
Yeah, I'll upload it. It'll be on websites and Zenodo, but as soon as it's uploaded and the individual slides, I'll send you a link, Mahdi, and I'll send it out to everybody whose details that I have if they've registered.
Perfect. Thank you.
Thank you. Thank you.
Thank you all for joining. Thank you, Lesley. Thank you.
Yeah.
Thank you.
And enjoy the rest of your day and your evening.
Good evening.
Thank you.
Bye-bye.
Bye.